irrigated there. We grow all kinds of rotations that include sugar beets, corn, potatoes, dry beans, all kinds of both barley and wheat, and a lot of seed crops, corn and carrots, etc. So our rotations are very diverse. We also have a very large alfalfa crop within the rotation as well. And so what I'm doing today is maybe just talk a little bit about just how important it is not to get stuck in our ways in agriculture, just how important research is. Research is critical to help us improve. And so when we get back out in the corn plots, I'm going to show you some examples of that hopefully will apply to this area. Although we're not exactly the same as you are here, or we're probably very far apart from each other in a lot of things. There's a lot of similarities as well in terms of how we can improve over time if we let research lead, okay? And so behind me here is one thing we're evaluating in Kimberly with sugar beets. Sugar beets a crop, as you probably all know, got a big root, but it's also a crop that has a very extensive root system below that tuber, which is around the 30 centimeter mark. And so most of our recommendations in Idaho and also other areas in the United States are all in the maybe top 15 to 30 centimeters of the soil. That's where a lot of our particularly phosphorus, potassium recommendations come from. But we are evaluating just how important the second foot is with regard to those nutrients in sugar beet production because of that type of architecture of the root. And we're trying to understand the interaction between the crop and different rooting depths and how that works, okay? And so in Idaho, we've learned that historically we've been sampling down to 3 feet for nitrogen, inorganic nitrogen for sugar beets. And our research indicates that that third foot isn't very critical. It's all the action takes place in the top 2 feet predominantly. 80% of our water extractions from that depth, although we do know sugar beets extract water down to 8 feet at least. And I'm sorry, I'm going to go between metric and empirical, but I can't really help myself, so I'm sorry. But it goes quite deep, okay? But 80% of the action in terms of nutrient uptake and water extraction happens in the top 2 feet. And so we're thinking about maybe even reevaluating and how we recommend soil sampling there. That maybe that third foot, which is harder to get to, costs more money to sample, may not be that critical. So we're thinking about adjusting from 3 feet up to the top 60 centimeters of soil. And so that's what this demonstration was to show that. You can look at that if you want to. We have phosphorus at different levels. We have phosphorus on top 30 centimeters, bottom 30 centimeters mixed between the two. And in Idaho, we found that when we have added phosphorus in that second foot, we have much better growth of the sugar beet, better water extraction as well. And so that's just kind of like a basic study we did this year to evaluate just how important that second foot is in sugar beet. And so that might lead into some future research studies in the field and maybe some changes in management practices as well. The sugar beets are pretty small right now, and I can't really see much over there, but that's just to demonstrate that fact that looking at that second foot for some of our immobile nutrients like potassium and phosphorus may be something we want to do with a sugar beet type crop. So anyway, yes, go ahead.
Oh, I was just going to ask, how do you plan on getting the nutrients that deep?
Well, we utilize, I started doing strip tillage work with sugar beets back in 2010. And with our units, we can put shanks on at different depths. And so you can actually inject fertilizer to whatever depth you want. And the hope would be is you would do that over a couple, 2-3 years to get that level built up down there. And then you wouldn't have to be doing that every single year. So you could actually put that big shank on your strip till unit, put it in, and then pull that off later if you want to as well. So that's some thoughts we've had with that. Anyway, any other questions at all about that? Well, so we do a lot of basic research. So we're all internally funded. So the federal government pays for all of our research. And so we're not chasing grants to the degree the universities are down there. And so we can have long-term studies and look at some of these studies that maybe aren't really that sexy or bringing money in at the time. And so we can look at things over the long term and doing fertility work, things like that, which is kind of hard to fund sometimes. It's something we do in our long-term studies within ARS. And so that's a little different model than you typically see in the university system in the United States. But I think it's very productive as long as you're doing research that has real impact and can change things. And so when we get down here, I'm going to show you some things we've done with that long-term strategy that have changed the entire dynamic of nutrient recommendations in the western U.S. So from our research.
Quick question.
Yes. Behind you there.
What's the kind of system for making sure that you guys are doing the right research and picking those research projects?
So we work under what we call research projects within the United States and ARS. So we have different areas that we work under. And at my research unit, we have an irrigation project. So I also, besides being a nutrient management specialist, I'm also an irrigation researcher as well. So I do a lot of work with developing crop water production functions for models. But we work under different projects, and those are 5-year plans. And we write up an entire plan of what we're doing, detailed. It gets reviewed externally by experts, both at the university system especially, and then we get evaluated based upon how good that research project looks over the next 5 years. And so every 5 years, all of our research projects are evaluated extensively and edited as needed.
Cool.
So, question here?
Yeah, I was kind of curious about your soil testing system. Is that state labs or is that more private labs that are doing the testing? Because you need one thing to analyze a soil sample, but it's another thing to calibrate them to make fertilizer recommendations. If you do a change, like such as going from 0 to 2 foot versus 0 to 3 foot.
I agree. In the United States, it varies. In the western U.S., in the Midwestern, it's all based upon private laboratories. North Carolina has a state laboratory, so their growers there pay a tax, and their lab is funded by taxpayers. So all their soil samples typically go to a state lab. But whether you're a state lab or a private lab, there's actually 2 very large proficiency programs. Utah State runs one of them, and Colorado State runs one of them. And so they send off a known sample to these labs, and they get results back. And then if they are doing a good job of analyzing the samples according to those standards, then they're certified by those proficiency programs. And so most labs will be in a proficiency program, and they'll be tested blindly throughout the year.
Yeah, but it's one thing to verify that your analysis is correct.
Yeah.
It's another thing to translate that analysis to a fertilizer recommendation, and that's sort of like making sausage. You don't want to see how that.
And that's done regionally. So, for instance, we in the western U.S. we typically use Olsen phosphorus extractable P. As you go farther east, they use a malic extraction. So all those calibrations are done locally in the area, depending on where you're at. And so all of our crop recommendations, for instance, for phosphorus, are all based upon Olsen phosphorus. So 12 inches in Idaho, yeah. Some places it's 8 inches, some places it's 12 inches, some places it's 6 inches. So I don't know how it is here, but in the United States, things tend to run by state borders. Even though it shouldn't, it does. Nebraska and Iowa have completely different recommendations, even though they share a very general growing area for most of their 2 states. So that's one thing they're trying to get away from in the United States now, is get away from those state boundaries into more actual agronomic areas that are very more similar than not. So anyway, okay. I think we're maybe ready to move over to the field. I got some information. We go look at them. About 400 pounds, 450 kilograms per hectare of phosphorus in the top 30 centimeters. This one here is the phosphorus is in the second foot, not the first foot, and this is mixed. The treatment for is mixed between the two different depths. And the one thing that we notice is that this particular treatment, where you have phosphorus added to both the first and second foot, is the best treatment in terms of growth. You can't really see that now, but in our columns too, we had 3-foot columns, so we had a 3-foot different 3-foot depth instead of a 2-foot depth here.
What's that tool for?
This is something they just gave me. It's like it's a shoehorn, I think. I'm using it as a pointer today, though. It looks like it's got a sharp edge. I probably, it's nice. You don't have to bend over, you just stick it on. Yeah. Now for other crops, this may not be as interesting. So like a lot of our cereal crops, they tend to have a fibrous root system, and it makes really good use of the top foot. And so we suspect that this second foot fertility issue will not be as much of an issue with those types of crops. But when you have a taproot type system, it might be more advantageous to think about what your second foot has in it. Because sugar beets has a very high phosphorus requirement. And based upon my research, when I did different rates of phosphorus, I never got the yields to kind of taper off. They were just as a linear, just kept going up and up and up, irrespective of how much phosphorus I put on. And it may be because that top foot, the sugar beet may be less efficient at utilizing phosphorus in the top foot, but that's where all the recommendations come from. But with a big fraction of the roots in that second foot, or that 30 to 60 centimeter depth, increasing those phosphorus, potassium, immobile nutrients at that depth could potentially have a positive growth response to that. And so that's what we're evaluating. I can't say that yet, but our initial study in the greenhouse with these tall columns is indicating there's something there that we need to evaluate further in the field. So we'll continue to do that over the next probably 5 to 6 years to evaluate that particular concept. So I had a hard time getting away from PowerPoint, so I did a hybrid. So how do you do a PowerPoint in the middle of the field, right? Well, you make big posters like this so you can look at them. But what I'm wanting to do now, you see behind you, you have a nitrogen study on corn, and I'm going to get to that in a second because in my part of the country, one thing we have to do, and we have not done yet, is get good recommendations of fertilizers for corn. Corn is fairly a new crop in the Northwest U.S. We had about 500,000 dairy cows move in the last 20 years in a 5-county area. So corn production is fairly new for us relative to other parts of the country. And we don't have any recommendations. We have a recommendation, but it's from 1970 data on sweet corn. So what we do need is we need this type of study in our area. And I'm going to show you just how much we do need that in a second. But again, I don't need to point out where I'm from yet, but what I'm really wanting to do here is talk about improving nutrient recommendations in irrigated production systems. So we're basically fully irrigated where we're at. Okay, we have very little dry land production. And irrigation's nice in the sense that it kind of takes the major yield limiting factor out, and that is water. When you're short of water, it doesn't matter how much nutrients or you do or do not have in your field, you're going to have problems with regard to your production. But with irrigation, you can minimize that. However, it's a lot of work to manage irrigation. And a lot of my work as well is trying to determine how much do we have to irrigate and how can we manage irrigation, particularly during drought periods. And so I'm not going to talk much about that today, but I'm going to go to the next slide here. Maybe I'll tear these things off of here. There we go. So what I would say to you is, when it comes to research, it's really important to allow research to take place and to not only think about research in the short term, but to think about it as a cumulative data synthesis. And so we found a lot of value in not only looking at what we're doing now, but what did they do back in the 60s and 70s? I'll hold it right here. In the 60s and 70s, and looking at data in more of a cumulative approach and how valuable that can be. It's really important that we have organizations that are doing agricultural research, because we've done a lot. We've been able to improve our systems, our economics, our food production over time through good research. So I would say this in terms of what do you do? Well, you follow best management practices. The ones that exist now, follow them. But don't get so stuck in those that you're not willing to expand your mind and think about what can be next. How can we continue to improve? Producers and growers that do that will be way ahead of the curve and be able to handle a lot of the changes we're going to be facing in the future. So let's go to the next one here. I want to share with you a couple of 3 examples of what I'm talking about here. And these are examples of how things can change even today, even in the environment we live today, where we've had lots of improvement within the agricultural complex. I want to talk to you a bit about some of the sugar beet recommendations we've adjusted. I want to talk about some issues we have with corn silage recommendations in the Northwest U.S. And also talk to you about some things we learned from manure and how that's changed our systems, which we did not expect to happen. I'm going to show some data on that, okay? And maybe as you go through this, think about your production system here and how things are changing and how, I think it's the right one, yeah. There we go. So this is the area that we're talking about here for sugar beets. This is about, we cover basically the Snake River Plain in southern Idaho. We also have parts of Oregon and Washington as well. And so you can see the sugar beet growing areas across the United States, concentrated in particular areas. You also have one here as well, up here in this area. I think Tabor is where is that where the sugar beet factory is? Yep. And so we always have people from Alberta coming down to our sugar beet meetings, and I know several of them very well, so really good researchers here. Okay, next slide.
Jay Anderson, one of them?
Yeah.
Yeah, you mentioned.
And Peter Retnig is another one. You know Peter? Yep. So this is the issue we have. This is our sugar beet yields across that growing area, northwest growing area. And this is in metric tons per hectare. You can see that, you know, from the start of sugar beet production in that area up to about 2006, we were increasing about 0.55 megagrams per hectare per year, okay? Then look what happened in about 2006. It went from 0.55 megagrams per hectare per year increase up to about 1.5. And why do you think that happened? Roundup Ready and neonicotide seed treatments. We are now on average over 90 metric tons per hectare, okay? And the problem is this. Our nitrogen recommendations in the past, from about last year and before that, were based upon yield. They were tied to yield, okay? So what we did is we took, I think back in the 1970s, there's research that said that you needed about 4 kilograms of nitrogen available, either the soil and fertilizer, for every metric ton of beets. That's how much nitrogen you needed, okay? Now as yields drastically increase, that means your nitrogen recommendations are going to skyrocket as well. The question is, do you need that much nitrogen to raise beets, right? And so from 1970 to 2022, that's what we did. You multiplied how many tons of beets, or how many metric tons of beets you're going to grow, and you multiplied that by a factor, okay? Now what we found was, so as your yield goes up, your end supply has to go up. That's how it's linked together, right? It's called a yield-based approach for nitrogen recommendations. Well, I got 2 of them. Oh, I do. This graph looks really busy, but this is kind of an example of, I want to show you that if you take into account all the research data going all the way back and see how it relates to the data you're collecting now, you can maybe see a more clear story of what's going on versus being kind of stuck in the moment, having blinders on, okay? Back in 1970s, the Amalgamated Sugar Company did some research, and they determined from that research that you need about 4 kilograms of nitrogen per metric ton of beets produced. That's what you needed available, okay? And if you were to apply that to the yields over time, this is what your nitrogen recommendation would look like. So if we were still using that 4 kilograms of nitrogen per metric ton of beets today, we would be recommending about 350 kilograms of nitrogen per hectare, which we don't need. Okay? When your yield is tied to your recommendation, this is what can happen if you don't have continuous research going on to tell you what's going on, what's happening. Especially when your yields are increasing so dramatically, as we're seeing down in southern Idaho, right? In 1996, the University of Idaho did studies. They determined that you needed about 3.5 kilograms of nitrogen per ton of beets, which is less than the 4, right? That's what they determined. And then this line here will show you that if we continue to use that 3.5 and didn't do any more research in the future, they would be recommending about 300 pounds today if that 3.5 was used. When I got to ARS, I worked at the University of Nebraska for 6 years before this, but when I got to ARS 15 years ago, we started a series of studies to see if this factor needed to be reduced a little bit. And yes, it did. In 2010, we found that at that point in time, you only needed about 2.7 kilograms per metric ton. And then in 2019, that dropped to 2.1, and then last year, 2.0. So you notice that this factor is going down, right? And what it's showing you here is that when research is conducted, the amount of nitrogen we needed to maximize yield and quality was pretty stable. Look at that. If you draw a line across here, no matter what our yields were, whether we were yielding 20 ton, say 40, if we're yielding 40 metric tons back in 1980 to 90 here, we need about the same amount of nitrogen. So it's what we call a static approach. And so what we found out is that we changed our recommendations this year from a yield goal-based approach to a static range, to around 200 kilograms of nitrogen per hectare plus or minus 20 kilograms. Now without research, we would still been stuck back in these days, putting on 300 to 350 pounds, thinking that extra yield we're getting now needs extra nitrogen. Well, we found out we don't need that. We're much more efficient within our system now. We can raise 90 metric tons of beets per hectare with the same amount of nitrogen as I needed back in 1970. So, without looking back at the data and kind of see how it fits into the puzzle, we would never have came up with this static approach. Okay? Yes, question back here. Well, yeah, he's saying whether we did any post-harvest soil nitrogen testing to see whether or not those would influence the subsequent crop. In our irrigated system, we tend to see our fall soil samples very stable. Okay, we don't see a lot of excess nitrogen being carried over. Most of our nitrogen is mineralized in the spring, so we tend to see a big influx of nitrogen from the fall to the next spring going up. But we typically in our management systems, we only sample in the spring for recommendations. And so our spring soil test is what we tend to use to add into this recommendation of N supply.
But your soil test, you're testing. Oh, sorry.
Okay, yeah.
If you guys were following that 350 recommendation, you would expect that.
You probably would have more residual in your soil. Yes, you probably would. Yep. And that would reduce your inputs in the future.
But your soil testing you're doing is for the beets. You're not soil testing beets. You go once every 4 years, right?
No, we soil sample every single year for nitrogen.
So the samples the year after the beets in the spring were not anything different than all the other fields.
Well, it's a very complex system because these beets are typically in a 3 to 4 year rotation with other crops, and so and all of our recommendations take into account nitrate and ammonium nitrogen to at least 2 feet. And so irrespective of what happened the year before, excess nitrogen being applied, residue management, they only recommend based upon soil test ammonium and nitrate. And what I'm going to tell you right now is there's more to the system than that. We're finding in corn, our spring soil test is not telling us anything about how much nitrogen we got to be applying to corn. So I got a slide here, I think. Is that the next one, maybe? Oh, not yet. Anyway, I'll show you in a second that we feel, at least in southern Idaho, our soil tests we do in the spring for corn is not going to help us predict how much nitrogen we need to supply to that crop. Because we're not getting much response to nitrogen that the current recommendations are saying we need. And so we have to come up with something different. So whether it be a pre-side dress nitrogen test or some other method to help us guide our management, because right now, just depending on a soil test is not going to cut it with corn, for whatever reason. I'm not sure if I answered your question very well, but I can't hear you very well. But if we want to talk a little bit later, we can about exactly what you want to know. I don't really think I quite understand exactly what you're asking at this point, so we can talk about that.
I got a question about sugar.
Yes.
You guys check the sugar, like, because that's what they growers get paid on this amount of sugar, right?
Yeah, our sugar percentage has increased, so we're running around average between 18-19% sugar.
So didn't that high levels of nitrogen?
Yeah. What's going to happen is if you drastically oversupply nitrogen, you tend to get really good yields, but your impurities and your quality drops, which then and your sugars go down as well. So it's kind of double whammy. You get a sugar, your total sugar may be higher, but when you have impurities, it's harder to get the sugar out of the beets, and then they dock you significantly in terms of your payment because of that. So with high excess nitrogen, you're going to have issues with that. Yep. But this next slide shows you an interesting factor here. So this is 1,000 fields between 2016 and 17, showing you what the farmers actually applied. Okay, and you can see this red line here showing you where our research shows it should be at. Everybody is over here. 53%, 54% of the fields are applying in excess of 100 extra kilograms per hectare than they need. And that, with current fertilizer prices, is about $341 per hectare. So even in our area, where you have a grower base that's pretty interested in this type of stuff, they're still way over applying fertilizer. And this data is from 2016 and 17, showing you how much because back then they were utilizing that yield goal-based approach. They were using that 4 and that 3.5 with these high yields. They hadn't dropped back and adjusted yet, and this is the result of that. It's way over application of nitrogen fertilizer. Significant economic and quality processing issue in the factories as well. So this shows you how much improvement we can make when we look at not only the current research, but we apply and look at it. How does it fit into the puzzle with respect to the past research as well? So that's one example of kind of just how important research is and what kind of advancements we can make. So this static N approach is new this year in the grower book in Idaho. Okay, I did these sites. These are all different. These are very large research sites. Corn plots, probably about. I'm guessing maybe I can't convert it to. You guys, I get confused.
Acres is fine too.
We publish in metric, but we speak in standard units. So it's the worst. You do. I heard him talk about bushels over there. Okay, good. You guys understand me. Perfect. Okay. So these are probably we're talking large plots on producers' fields. And as I said, silage is new to us. We haven't had a lot of experience with it. Okay, so what do people do? They put it out there, they put a bunch of nitrogen on it. And these are dry. These are actually dry. This is no moisture. So these yields look terrible, but that's because there's no moisture in them. You guys don't talk about yields with no moisture in them.
Way too low that way.
I know. And that's just how we publish them. So I'm sorry, I should have had them standardized to some moisture content, but I don't. But the important thing is this, these are all rate states. We went from a control, like this is a control plot here, and we increase our nitrogen. As you go across these plots here, you go from low nitrogen to high nitrogen, okay? A nitrogen trial. Now, we got no response to nitrogen, even though the recommendations we have now said we should. So this is what the Pacific Northwest nitrogen supply recommendations were right here. And this is our lowest end supply. So this is the supply we had just in the soil without no fertilizer being applied. And you can see that some of them are close, some are a lot lower, some are kind of in various ranges. But we saw no response to nitrogen. That means my yields in the 0 N rate was the same as all my other rates.
Well, that's because you're growing after sugar beets, all that excess nitrogen.
Well, these are also after small grains. Our rotations are very diverse. After alfalfa, and it could be some of that, right? We also live in an area where there's lots of dairies, and we have data now from our research location that you're seeing deposition between 10 and 40 pounds per acre of ammonium being deposited across our growing area from those dairies.
Atmospheric deposition.
Atmospheric deposition. In the areas close to dairies, it could be up to 50 pounds per acre. It, on average, across the whole growing area, downwind is about 10 pounds per acre. So it's adding nitrogen to the system, right? You're also coming after alfalfa, perhaps, after maybe a sugar beet crop where half the nitrogen ends up in the tops, half ends up in the bottoms, okay? So when you think about sugar beets, we know that about 50-50 split between the tops and the tubers. So, and that's readily available nitrogen, about 100 pounds of nitrogen being deposited back in the soil. But we do know we need nitrogen in many of our systems, but we don't know how to predict it. And so we need studies like this right here to help us figure that out. So it goes to show you, just because you have a recommendation doesn't mean it means much, especially if there's not much data to back it up. There's not much data to back these recommendations up. And we haven't done the studies yet because it's not sexy research that people want to fund, right? And so you need people that are willing to buckle down and do it because it can have a huge economic impact on the grower area. Okay, last example here. This is an interesting one here. Thank you very much, Ken. So when I worked in Nebraska, I was a soil fertility specialist there. After I started my career there, I was there for 6 years. And all the fertility people, all the soils people got together and we revamped all the recommendations for corn in Nebraska. And one of our protocols was this, is we had to, you have a research study on a site that had been manured in the past with cattle manure, it had to have been at least 4 years before, because our thought was, based on the recommendations, after 4 years, the thing's back to normal. It's back to some baseline, that manure has burned itself out, whatever you want to say, right? That's kind of what we thought. I hear some naysaying about it. I'm going to tell you that's what you're thinking is true. That is not the case. But if you look at the University of Nebraska's even current recommendation for how to credit manure over time, they give you basically a 3 year, 3 years from now, you can give that nitrogen you applied about a 5% availability, okay? And after that, it's kind of went away, it's gone, okay? I'm going to show you that is not true. And I want to show you some interesting data we determined over a long-term study. So in Idaho, as you know, we have lots of dairy manure. And so we put 3 different rates of manure on, all the way from 0 up to 238 megagrams per hectare, okay? Pretty significant application. We did that for 4 years, from 2000, I think it was 2004 or 2009, okay? We put this on. Then we stopped applying and kind of farmed it conventionally, okay? And part of it can be farmed conventionally without any added nitrogen fertilizer. Let me show you some results of this. This is a sugar beet crop. Can you see any difference in the growth in there? Now this is 2016, so it's 7 years after manure stopped. For 7 years now. You can see the early growth of sugar beets where we put no manure and where we have manure at 2 different rates. Drastically different early growth response to that. And this is 7 years after with no added nitrogen to it. Now, the interesting thing is this, the 0 here, this has nitrogen applied to it. Fertilizer. So what that manure is doing is above and beyond just the nitrogen it's getting from the manure. It's changing the system somehow. And we don't necessarily understand exactly all that's going on. Yeah, phosphorus is higher here, but it's adequate in these plots as well. Based upon recommendations, yes. Okay. Okay, let's go to the next slide here. And what I want to show you now is I didn't put the axis on here, but this is available nitrogen from 0 to on this axis right here. So this is ammonium and nitrate nitrogen from a spring soil test in the top 2 feet. Okay, and this is 14, 16, 19, 2020, and 2021, all the way up to 12 years after manure application. And these are on the plots, the parts of these plots that have had no additional nitrogen fertilizer. And you can see that that manure, even 12 years after, is still supplying nitrogen to the system compared to the fertilizer.
But you said earlier you put 4 years, manure on for 4 years in a row.
Well, the 238 was every year for 4 years. The lower rate was every other year during those 4-year period. That's why you had the lower rate. So the annual rate was the same. It just applied only over 3 years versus 2 years over versus 4 years. And you can see that with the highest rate of manure applied every year for 4 years, on average, it's giving you about 225 additional kilograms per hectare, even 12 years, across all these years. And so that manure is more than just 4 years. It's up to 12 years of supplying nitrogen, okay? Let's talk about yields now. What we're getting from that manure compared to no manure. You can see that in 2019, 10 years post-manure, 2020, 11 years, 2021, 12 years after manure, that we have corn silage, barley yield, and corn yield, that we're seeing a advantage in terms of yield increase when manure was applied compared with no manure was applied.
That's 100 tons per acre of manure you're putting up. Is that correct? 238, 240 per hectare?
You're making me have to convert now.
I'm sorry, I'm still imperial. I've got gray hair.
Well, I have a hard time.
So 238 kilograms
This is metric tons per hectare.
per hectare, which is basically 100
Yeah, about 100. Yeah.
for 4 years.
No, total, cumulative.
Cumulative over the 4 years.
Cumulative, yeah, so these numbers here are cumulative over the 4-year period. So that's the total amount was applied in that 4 years. Yep. So yields are still increased with that manure application. And this is looking at so I said before that it was more than just nitrogen that's going on here, right? Because we did some, we know that the yield increase is more than just nitrogen being made available. And this is the microbial activity. This is looking at 4 different enzymes that are responsible for the cycling of sulfur, phosphorus, and nitrogen, okay? And you can see that this is 13 years post-manure, sample in the top 6 inches. We're finding that when you have manure applied, and this is 0 to 6 inches, 6 to 12 inches on the bottom here. When you have manure, you increase the microbial activity even 12 years after the application. So it's changing the entire microbiome in our soils. It has a long-term effect, and we don't know how long it's going to last. And so what we're doing here in our research locations, we have four different individual studies. They're going to be, we've had manure applied, no manure applied, and we're going to look at those effects from now until whenever they want to stop the studies. And so they're long-term studies to understand the impacts of manure application on production, fertility, how it's going to change fertility. But all I can tell you is when you put manure on, you change the whole system for a long time.
There was some research done here. Dan, I don't know if you're familiar with the manure application they did like 20, 30 years ago.
Oh yeah, this
this
was the 50th year this year.
And
So
they still saw results after 40, 50 years.
it was discontinued in '86, so we've got a study just up the road where we've got a similar thing where application, some was discontinued in '86 and another one in 2006, I think.
Yeah.
So it's a long term, and it's for doing other research and having manure on your plots, don't let it happen.
Yeah, it basically is a whole different system. You can't just put manure down and expect it to go back to what it was like before anytime soon. And based upon this, it can be decades before that happens. But from a production standpoint, there's some benefits here. Potentially long-term benefits of utilizing manures. And we can't look at it like we did 20 years ago. And so we have within ARS across the entire country, we have what we call a manure priming project, where we put manure down either one time or over a short time, and we're evaluating that over the long term of what that impact of manure is compared to conventional farming. And so that started several years ago. What you're talking about what we call manure legacy. So any plots that have manure in the past, we're going back to those plots and seeing, okay, is there still an effect here or not? And how long is it lasting? So kind of looking at it from 2 different approaches.
And that's the other thing, we've got studies at the Ag Canada station, where they're looking at remediated land that was artificially eroded. And manure is amazing what it can do. Way more than any other amendment, way more than high doses of inorganic phosphorus. There's something about that and interacting with the calcium carbonate in our soils that is exposed when you erode those soils away. So it's something I think is a bit underappreciated. People say, why are you doing manure work? You know, Pliny the Elder knew this in ancient Rome. No, I don't think so.
Yeah.
You don't quite have an answer.
So this is maybe just a few examples that I had that indicate that we have definitely not figured everything out. There are things in the past 10 years we figured out that we thought we knew we don't know. And the important thing is, I think in the agriculture world we all live in, is that research is critical, but it's how we do research is important, and how we analyze data is important. We can't just look at this study over 4 years and say, okay, this is how we're going to do things. You take this study from 4 years and you look at all the previous data as well and see if it tells a better story when you analyze it as a whole. And so that's an important concept that we've learned that's changed everything in Idaho for us, is that type of an approach. So although I'm not from here, I don't understand your systems very well, hopefully there's some things we learned there that can apply to you here in southern Alberta. So anyway, I was just going to, I don't need to show these other slides, but I'm going to show them anyway. So this is an iceberg. This is one of my favorite pictures. It doesn't look very good here, but this is a picture up in Greenland. You can see a diver over here of an iceberg, right? And what we know is a small fraction of what we don't know. And if we want to do better, we have to uncover more of this iceberg. And the only way we can do that is through research. And so what I'm showing you here is all the studies we're doing. This manure thing is so critical to us because we have so much manure, and transporting it is so crazy in terms of cost. We have to understand the value of it. The more valuable that manure is to a producer, the farther we can transport it. And it's one of the objectives of our priming study, is trying to figure out what is the real value of this stuff. If it's really valuable, then we can transport it a lot farther, okay? And so we have all these studies going on now at just our location to understand the long-term effects of manure. I got one last slide here too. I'm not sure if it's important or not, but I'm going to show it anyway. Anyway, this is already what I talked about. Just how much we need data, synthesizing cumulative data, etc. But if anybody wants to contact me, I have a copy of these slides, and I'll pass them out to you. So if you want to, my contact information will be on those. So I'll just pass that out when we're done, and you can pass that around. It has all these slides on there and my contact information if you need to talk to me. But anyway, this study here is something that's hard for us to get funding. The University of Idaho has a hard time getting funding for this, doing this work. But we need a way to manage nitrogen in corn, which we don't have. And so we've got to spend some time figuring this out in Idaho. So I believe in this particular study here, you got a control plot, and as you go down, you have increased rates of nitrogen. So they'll be evaluating that here. So anyway, I've talked myself out, I think.
Fantastic. Is there any other questions for David?
A question right here.
You mentioned manure. I've seen people in southern Ontario. They get manure from city.
Yeah.
Dry them, and do they work too?
You know, I haven't evaluated it. We haven't spent a lot of time understanding differences between different manures. I do know that they're showing big advantages for swine manure, poultry litter, having long-term effects as well in other parts of the country. But I would suspect that anything that changes the soil microbiome in terms of manure, you're going to see positive benefits over the long term. I wouldn't suspect there's a whole lot of differences between, there's maybe some supplemental differences between cattle and dairy. But I haven't looked at that myself, but I have heard that other manures are also showing a similar thing as well as we're talking through this whole process. Yep. I kind of look at it like this. I mean, I look at it, at least in my mind, I like analogies, right? So I'm thinking of when you add manure, that soil microbiome is going to be changed for a long time. It's much more complex. It's doing a lot more. It's contributing to yield in ways we don't understand. And so one of the things we're trying to do now is we have a consortium of ARS soil microbiologists, like 30 of them. They now have a group formed. And their sole goal is one of their objectives in the research project is trying to find out what are the changes and how are those changes affecting crops? You know, when you go from like a small town to a metropolis, that's how kind of equivalent to when you before and after manure is the microbiome is much more active. It's doing a lot more than it was before. But what is it doing? What can they measure? And how can they relate what they measure to what's actually being produced? And so they have a huge number of microbiologists trying to figure that out right now on some of these plots. So.
Sounds like a pretty shitty group to be part of.
Oh yeah, real shitty. Yeah. Yes.
Just going back to kind of what I had asked Ben, like with that 200 kgs a hectare or whatever that kind of maximum end rate was, basically by bringing down how much you need per unit of yield kind of thing, is that going to continue, you think, if we try to keep increasing yields in some of those crops, or does that number change? And is that like, why is it that number specifically, and have you kind of figured that out, or is that just kind of the buffering kind of capacity?
At this point in time, it's just an observation. We know it's real because we have all the past data, and we can say it's a real thing, that around 200, plus or minus a little bit, depending on what texture, the sandier your soil is, the more you're going to need, etc. But what we're doing is we know that in the future, if yields keep going up like that, we're going to have some studies to just check this and make sure, is that 200, is that right? Do we need to go a little higher? So we're going to keep an eye on that over time. So we're committed ourselves, myself and the sugar beet cooperative and their agronomists have decided that every so many years we're going to do some check studies to make sure that we're not, if yields keep going up, that we're still where we need to be at. That needs to be evaluated. We can't just rely upon that without any further research.
So all that excess nitrogen, where did it go? Because you said it didn't show up in the soil test.
Well, that's a great question. Let me give you another scenario. So to show you how I don't know, right? So we did some studies looking at trying to minimize irrigation water usage. So we had these rotations. One of them was a fallow in there. So we had all these rotations, and we had one of the treatments had a fallow in there. And so they would sample. We sampled the soils in the spring, had a certain level of nitrogen, right? All about the same. We fallowed where we had a crop growing. We fertilized as well. Went back in the next year, and the fallow had higher nitrogen than it did the spring before, and it was the highest compared to all the other fertilized treatments as well. Where did that nitrogen come from? Yeah, mineralization, but significant mineralization, right? We do know that in our part of Idaho, even back in 1960 when our location was opened, the first soil scientist there, he made a statement in a paper in 1963, I believe. He says, for whatever reason, we can't, because our organic carbon contents are around 0.5% to 0.8% organic carbon, so we're not a high carbon system, and so people didn't think we could grow crops there. But we grow really good crops there, high yielding crops. But he stated that for whatever reason, these soils are a high mineralizing soil because we get a lot of mineralization in low carbon soils that we don't necessarily understand why that's happening. And you don't see that in all places either.
Well, you know, Ross McKenzie did some work a number of years ago here on irrigation, non-manured fields, and found between 40 and 160 pounds of nitrogen.
Yeah. Yeah.
Where did it come from?
Exactly. And that may be one of the reasons why we're not getting a response to nitrogen in our corn, is because we have a lot of nitrogen being mineralized that we can't account for. And I would say it happens after their spring soil test. You know? So we're not necessarily capturing that whole system by a spring soil test. And so by sampling in the spring, that's our best management practice right now and for many of our crops. But the question is, should that be our best management practice going forward? Are there other things we can do to better match crop needs with fertilizers? And the question, I guess your question is, we just don't know where it all goes or where it all comes from. We know we're not leaching it very much because we're in a very deficit. So we have a hard time keeping up with ET in the peak of season. So we're not moving a lot of nitrogen down deep. We have very deep silt loam soils, and we do a lot of like in my area, we do a lot of measuring of soil water content down to 10 feet. And we're not seeing a significant amount of movement down below that. At least in our, there are some soils that are very shallow on fractured basalt that are losing a lot of nitrogen, but most of our soils are not, I don't think. It's getting captured in that microbiome somewhere. They're getting blown off as nitrous oxide a little bit here, a little bit's getting incorporated into more complex organic matter fractions, etc. So, but the question is, I always think of soil science and nitrogen management as the big black box of soil science. We just don't know what's going on in that box. We're managing around it best we can, trying to eat away at the box to get a better view of what's inside of it. But most of what's happening with the nitrogen cycling is in that black box. We just don't know what's going on. The more we understand, the better we can do, I think. So.
Well, I appreciate you coming, David. I know that our irrigated sector isn't really highly supported through the type of research and science that we probably need, so we'll have you back to come talk to some ministers and advocate on our behalf as well. I found that you can't advocate for yourself, you have to find somebody else to do it for you. Yeah. So maybe we can trade.
Yeah.
Yeah. And on the just one other thing, I'm not sure if you're aware, but there is a company out of Saskatchewan that we work with often that uses these PRS probes.
Yes, yeah. Yes, I know Ken.
Okay, sounds good.
Yep.
Everybody knows Ken. Yeah.
He's just as prevalent down in the United States as he is up here too.
A good
He's a part of a soil fertility work group that we meet every year, and he's there, so I know him really well. Nice guy.
Anyways, everybody join me in thanking David for coming up and talking.
Thanks everybody. Thanks for the shoehorn too.
No problem. Here's a
oh, thank you. Well. Appreciate that. Thanks, Ken.